Hello everyone, I'm Gayatri from first MSc Microbiology, Sacred College, Tirupattur. Let me discuss about classification of virus. Of virus, they are classified in the basis of host, structure, and nucleic acid, also based on genome nucleic acid like DNA, RNA, which is single standard or double standard, and sense and method of replication determined its classes. Other classifications are determined by disease caused by the virus and its morphology. There are DNA and RNA based on standards. It is classified into seven main groups. The major difference between viruses are a plant virus and an animal virus. Plant virus which infects plants and animal virus which infects animals and humans. Most of the plant virus possess a single stranded RNA. Animal virus possess a double stranded DNA. They are helical in nature. Animal viruses are a symmetrical form that can be seen. Plant virus transmitted to the plant sap by different vectors such as insects and nematodes and through a pollen horizontal transmission. Transmitted in many ways like respiratory tract, sexual contact, zoonotic disease, vector transmission. Spread from parent plant to offsprings through the vertical transmission. Some animal virus and plant virus are organogenic virus. Here are some examples for plant viruses: tobacco ring spot, watermelon mosaic, barley yellow draft, potato mop top, citrus citrusy, sugar beet curly top, etc. Animal virus can also common diseases like smallpox, herpes, measles, rabies, chickenpox, chikungunya, etc. Based on structure of virus, structures are classified into three types: helical symmetry, isohedral symmetry, complex symmetry. Helical symmetry, which is coated with protein and nucleic acid arrangement helix. Tobacco mosaic virus, rabies, influenzae, para-influenzae, mumps, measles. Isohedral symmetry, which is like polio, adenohepatitis, which a protein subunit covers the nucleic acid core. Complex symmetry, which is pox virus and bacteriophages. It is a helical symmetry which has protein and nucleic acid arrangement of helix, tobacco mosaic virus, and rabies, etc. It is a isohedral symmetry which is coated with protein, covers the nucleic acid core like adenovirus or poliovirus, etc. It is a bacteriophages which has complex symmetry, which contains the head, column, tail, long tail fimbriae, photoplate, etc. Into seven main groups, which is first classified by Baltimore classification in 1971 based on the standard and single stranded or double stranded sense and mode of replication. David Baltimore is a Nobel Prize winning biologist, classified the virus on how the mRNA is produced during the replicative cycle of the virus. Virus contains a double-stranded DNA, single-stranded DNA, double-stranded RNA, a single-stranded RNA with positive polarity, single-stranded RNA with negative polarity, diploid copies, which is two copies ssRNA, and partially double-stranded DNA in genomes. Positive polarity means that the genomic RNA can serve directly as an mRNA, and negative polarity means that there is a sequence is complementary to the mRNA. Here, which is group 1, group 2, group 3, group 4, group 5 and group 6, group 7, which is classification of virus based on single stranded and double stranded DNA and RNA, which enzymes like reverse transcriptase enzymes, and finally converted into mRNA. Let us discuss one by one. There are seven main groups of virus classification. Group 1, virus contains double-stranded DNA as their genome. Their mRNA is produced by transcription in same way as the cellular DNA. Examples like adenoviruses, herpes viruses, pox viruses. Group 2, virus have a single-stranded DNA as their genome. They convert their single-stranded genomes into a double-stranded DNA intermediate before transcription to mRNA can occur. Examples like parvoviruses. Group 3 viruses use double-stranded RNA as their genome. The stranded separates and one of them used for the template for the Generation of mRNA using the RNA-dependent RNA polymerase encoded by the virus. Example, reovirus. Group 4 viruses have single-stranded RNA as their genome with a positive polarity. Positive polarity means that the genomic RNA can serve directly as an mRNA. Intermediates of dsDRNA, called replicative intermediates, are made in the process of copying the genomic RNA. Examples, coronaviridae, flaviviridae, astroviridae, picornaviridae. Group 5 virus contains single-stranded RNA genomes with the negative polarity, meaning that sequence is complementary to the mRNA. As the group 4 viruses, single double-stranded RNA intermediates are used to copy the genome and produce mRNA. In case the negative stranded genome can be converted directly to the mRNA. Additionally, full-length positive RNA strand are made to be serve as a template for the production of negative stranded genome. Examples, orthomyxoviridae, paramyxoviridae, rhabdoviridae. Group 6 viruses have diploid single-stranded RNA genomes that must be converted using the enzyme reverse transcriptase enzyme to the double-stranded DNA. The double-stranded DNA is then transported to the nucleus of the host cell and inserted into the host genome. Then mRNA can be produced by transcription of the viral DNA and that was integrated into the host genome. Example, retroviruses. Group 4 viruses have partially double-stranded DNA genomes and makes single-stranded RNA intermediates that act as a mRNA, but also converted back into a double-stranded DNA genomes by reverse transcriptase, necessary for the genome replication. Example, hepanodovirus. Thank you for your patience listening.